Passive transport isn't cutting it. I can't move ions uphill like this.
I'm energy. Note disclaimer for 2.8 mechanism of transport.
In this video, before we get started, just a brief warning or a brief debriefing, I want to say. In this video, we're focusing more on active transport here and the distinguishing features between the different types of active transport. If you reviewed our earlier videos as it relates to Unit 2, you'll notice that you've seen the same image before, and you're probably thinking, I don't need to review this video because I already reviewed sections earlier that cover the differences between passive and active transport. With the latest transport related updates, meaning Unit 2 updates, there is a focus on just understanding specifics as it relates to active transport, which is why 2.8 might seem repetitive, but there's more of a focus on active transport. If you feel confident in your understanding of passive transport, please feel free to go through the chapters and select the information that you need for your better understanding. Please note in this video, we're going to be covering passive and then active transport, just as previously indicated, with more specifics on the different types of active transport. Let's go back to passive transport and distinguish the features between passive and active transport just as a brief review. Now, in passive transport, we're going along the concentration gradient. However, in active transport, we're going against the concentration gradient. Because we're going against the gradient in active transport, we're going to require some energy that is known as ATP. With passive transport, because we're going along the concentration gradient, we don't require any energy. However, there are two distinguishing features in passive transport, and that's diffusion, aka simple diffusion, and facilitated diffusion. With simple diffusion, you're going from areas of high to low concentration, which basically means that you have high substances, think salt on one side, with low substances on the other side. Because of this, you are going to need to move those substances to the other side. For simple diffusion, it often comes in the form of simple gases, while facilitated diffusion needs some sort of assistance, and that assistance is known as proteins. Now, a specific type of facilitated diffusion, sort of, is called osmosis. Let me explain why I say sort of. This is why the little arrow here sort of looks like it's under facilitated diffusion, but not. Here's the thing. We do require some sort of proteins that are known as aquaporins. Those aquaporins in the process of osmosis allow for low solute to high solute concentration movement basis of moving water. So water is moving across the barrier, but we are moving from low to high solute concentration, which is different than facilitated diffusion in terms of movement of substances. So while they both require a protein and while they both follow under passive transport, osmosis takes a look at opposite solute to solute movement. And this is important when we're talking about things like tenacity, which was previously reviewed in section 2.7. If you haven't done so, please feel free to review that video, and you could find it in our playlist as well. Now, when we talk about active transport, remember we're requiring some sort of ATP, and this is important for things like sodium potassium pumps, which take a look at different ions that need ATP to move across a barrier. There are other ways to utilize energy as well in a process called endocytosis and exocytosis. Specific types of endocytosis include phagocytosis, pinocytosis, and receptor-mediated cytosis. Now, if you're wondering why we're not going into more detail, it's because we are later in the video. Feel free to review the chapters for more details.
So I was dating passive transport and he just has no energy.
I might have no energy, but at least I'm fast and smooth.
I want to ring the hypothetical alarms here. So when you're reviewing AP biology, you want to think of the sections as related to one another. If you remember when we tried to go over Unit 1 and macromolecules, we covered lipids, and then I talked about the plasma membrane, and then we went into Unit 2, which we talked about parts of the cell and the plasma membrane, and now we're talking about what specifically happens within that plasma membrane. For AP biology or college biology, a lot of questions ask a lot of you to compare and contrast not just passive transport or active transport, but to compare it as a whole. So that's why we're re-going over previous concepts that you learned before so you can remember the differences between active and passive transport. So when we talk about passive transport, we're talking about this idea that there is no protein involved right now because this is considered a simple base passive transport. When we are talking about that, we're talking about those small nonpolar molecules, often in the form of gas, and they don't require energy, so they're going to go through with ease. They're going along the gradient, which means that we're going from areas of higher concentration to areas of lower concentration, and basically we're going with the gradient. Now, if we compare that with facilitated diffusion, that involves a protein. So you'll see a protein that is involved and it allows substances to cross easier than without a protein and is often left for large charged molecules. And it is also areas of higher to lower concentration. So be aware that when you're comparing for passive transport and facilitated diffusion, that they both are high to low, but facilitated once again has a protein that is involved. And this is left for things like ions and glucose that need some sort of assistance. Now, note that there is no energy involved, so we are still under the category of passive transport. Remember, we talked about osmosis, we talked about water potential, and the idea that we're going from areas of higher concentration to areas of lower concentration. So if you remember, we're going from high to low in the idea of simple and facilitated diffusion, but osmosis, if you remember, is a topic that often can be confusing because we're talking about low solute to high solute. So I want to keep that in memory because we're going from areas to basically equalize the concentration of the solute. So when I'm talking about water potential high to low, you want to think we're talking about the water and not the solute. So let me break this down even further. So if you look at this picture here, you can see that there's a high salt concentration on this right side, which basically means that we need to move that water to the right direction because there is not a lot of water here. So we are talking about the water potential, and the water itself is going from areas of high water concentration to areas of low water concentration to basically equalize the concentration itself. So the water potential is what's important, that we're moving areas of higher water potential to areas of lower water potential. I want to also recap when we talked about our diagram, we're going in low solute to high solute. So let's go back to the picture here. When we're talking about osmosis, you can see here that there is low solute on this side and a high solute concentration on the right side. So with the solute, we're basically going in opposite to low to high. So this might be confusing, but when you're talking about osmosis, remember we're talking about aquaporins and we're talking about the movement of water itself, which is why when we're talking about high to low, we're really focusing in on the water potential, not the solute potential, unlike facilitated diffusion and simple diffusion, basic types of passive transport. So while this follows under the category of passive transport, you want to understand that we're focusing in on that solute concentration, which needs to be disturbed by moving that water around. To do so, we go from areas of high water potential to areas of low water potential. This diffusion involves aquaporins again and involves this idea of the diffusion of water along the selective permeable membrane. So instead of moving the solutes, which could be something like salt, we're moving the water instead.
Sorry, ladies. No one passes through this membrane without energy.
But we want to go against the gradient.
Then you're riding with me.
Let's talk about active transport. In that little clip right there, you saw the individual drink some sort of energy drink, and that was supposed to be synonymous with the term ATP or energy. Remember, you can't get inside the club or inside the cell without movement against the concentration gradient, which means that you have to go against the grain. So what you want to remember is, say you have a stream of water and say you have a leaf. Oftentimes the leaf will land on the stream of water and it will push it in the same direction. Now, if we want it to go in the opposite direction, we'll need some sort of outside energy, say wind or something that is going to push that leaf in the opposite direction instead of moving forward. And that's how you could think of active transport. We're moving against the concentration gradient, and we will require some sort of ATP. There is three main forms, which includes the sodium potassium pump, which we're going to talk about in just a second, endocytosis and exocytosis. Endocytosis can be further broken down to receptor-mediated endocytosis, pinocytosis, and phagocytosis. All of these require some sort of ATP, which we're going to conclude or talk about in just a second. Before we do so, remember these membrane proteins because they're important to note. Now you can see that there are integral proteins that are shown here, which means that they follow throughout the plasma membrane. The proteins can function as channels. So the channels are just like a tunnel that allows the different items to cross through. Carriers or receptors. So carriers would be the idea that they sort of move them from one side to the other. Receptors would be on the outside, in which we'll discuss in a later part of this video. Now let's talk about active transport more in detail. When we're talking about active transport, I want you to remember that we do require ATP. And there is this idea that we're moving against the concentration gradient. There are two main forms. Remember, we're going from low to high when we're talking about these two forms. The first one is primary active transport, and the second one is secondary active transport. Now, in the first one, we're talking about this idea that we have a pump, and you can see that pump there, and it's just this idea that we're moving 3 NAs for 2 Ks. This is called the NAK pump. And you can see it's one pump and one ATP here. And this is just to state that with primary active transport, you're using ATP pretty directly across the membrane, in which you're moving ions at the same time. So in this case, we have 3 Na for every 2 K, hence the reason why it's called an NAK pump. Now, you can have a secondary active transport, which is a little bit more complicated. So for example, you could have what's called a sodium glucose symporter. So this seems very complicated for students to understand, but you can think of this idea that you have this inside the cell, and inside the cell you have these pumps, such as the NAK pump that we talked about with the primary active transport, where you want to think of this NAK pump moving the sodium and the ions back and forth across that barrier. But at the same time, when they're moving the sodium and ions, so exists this glucose transporter, which will move glucose across the membrane. And so this is utilizing basically two forms where one pump is dependent on another. So the movement of one substance or these ions is dependent on other ions or solutes. So this is a tad bit complicated because it's not just using one pump, it's dependent on another pump, and the other pump basically works to move other items across that channel. So when we're talking about active transport, remember we're talking about ATP, and you're going to find that we're talking about this more in detail when we talk about respiration and photosynthesis. I want you to keep that in mind. So when we're talking about the formation of ATP, you might see pictures where they have an arrow designation. So an example of this I could show you is in this photo here. So you could see here when we go in, you have ATP that has been broken down into ADP plus P. So what happens is you have ATP, which stands for technically adenosine triphosphate, tri meaning three. So ATP technically has three separate phosphates. And then what happens is you'll see the arrow and it will say ADP plus P. Sometimes you'll see a little I indication there, but you'll see a P symbol. And what that indicates is adenosine diphosphate, which means two phosphates, and then the one broken up. So the act of ATP breaking down into ADP plus PI is the release of that energy. And that's technically what's happening when we are talking about active transport. We're not just talking about using up ATP, we're technically talking about taking that one phosphate off, and then membrane proteins will essentially use this energy to move the different molecules. And once again, they're moved against their concentration gradient. So it's from usually we're going from low to high, or if you look in the photos previously, we can take a look and see that we're going against the gradient.
3 na OUT, 2K in. That's the deal.
But we don't want to leave. It's against the gradient.
Let's talk about a sodium-potassium pump. So when we're talking about a sodium-potassium pump, we're talking about this idea that we have 3 sodiums for every 2 potassiums, or 2 Ks. The idea is to utilize an enzyme, which is known as ATPase. So if you remember in Unit 1, we mentioned that the ending ASE signals an enzyme. And an enzyme is not required, but assists in breaking down or formulating a reaction. And the way that it's helping out in this case scenario is by breaking down ATP. ATP, which means adenosine triphosphate, or 3 phosphates, breaks down into ADP, or adenosine diphosphate, which means 2 phosphates, and that additional phosphate is broken off. So that is how energy is utilized in this case scenario, to basically pump out 3 NAs into the outside of the cell, while 2 Ks inside the cell. And the purpose is to continuously do this to maintain a sense of ionic balance. And that's what is going to allow for what we call electrochemical gradient. And what that means is, if you remember on the periodic table, sodium is highly positive, and then also we have potassium. So because of this, the behavior between sodium and potassium, the idea is that it creates an electrical gradient because you have 3 sodiums which are out while you have 2 Ks coming in. So there's a net negative charge, or a negative 70 MV. Now, you don't need to know per se the exact voltage, but you should recognize that this behavior is really important for driving nerve signals, muscle contraction, and nutrient uptake. I want you to think of things like the heart, or when you have some sort of nerve issue, you often think of some sort of electrical gradient, and you want to think of it that way when you're talking about the sodium-potassium pump. This is really important for things like cardiac glycosides, which is just a way to say heart failure, dysfunction that leads to swelling or cell death, and energy depletion if there is some sort of result that leads to low ATP. So any issues with this naturally occurring sodium-potassium pump can lead to these heart-related, or I should say, issues throughout the body, including heart, pump issues, and energy depletion. This is important not because you are going to be expected to memorize the medical significance, but because you want to recognize this for a potential FRQ, which can relate a sodium-potassium pump or your knowledge of the active transport back to your understanding of what it could be utilized for. So remember that the sodium potassium pump essentially wants to create this overall negativity inside, and this is what drives the gradient, which essentially drives ATP production. Now, let's talk about the other two types of active transports, which include endocytosis and exocytosis. So if you remember, endocytosis is the process of engulfing, and you could see the root en or enter to form a vesicle. So oftentimes we talk about phagocytosis, pinocytosis, and receptor-mediated endocytosis. And you often think of this when you're talking about really large particles to go inside the cell. Enter, endo. Think en, enter. So N, you have phago, pino, and receptor mediated. So let's talk about phagocytosis. So phagotes are rather large. So we're talking about large particles, often solid, that will create this sort of pinching mechanism and engulf, creating a vesicle or a food vacuole in this case to bring the product inside the cell. Now, pinocytosis acts the same way, where it pinches off and it creates a vesicle. But if you notice, the particles are a little bit smaller. So pino, think pint, very small. Then you want to think of receptor-mediated endocytosis. So in this case, you'll notice that very small, there are these hooks. So there's a hook there, there's a hook there, and there's a little tiny one there, and those are your receptors. So they're often shown in books like a little hook, and what ends up happening is the particle themselves will basically bind to the receptor. And this signals to then create that pinching mechanism that you see on here in the pinocytosis and phagocytosis. The same thing happens with receptor-mediated endocytosis, where it creates a little bubble and then will eventually create a little coat or vesicle to then have the product inside. So you can see here, as it forms the bubble, inside are the receptors with the little particles on the inside. So that's what receptor-mediated endocytosis is. It's sort of like a key that attaches to a lock that signals to create this sort of vesicle and then sort of fold upon itself to then move products inside the cell itself. Now we talk about exocytosis, and when you think about exocytosis, remember you think of the word exit. So the vesicle is already formulated in this case, and then the vesicle formulates with the plasma membrane to then push out the product. So it's important, especially when we're talking about neurotransmitters that are basically signal from the nucleus to go ahead and release the vesicle related products outside the cell. And remember, earlier in Unit 2, we talked about like the Golgi apparatus and the function of that and how that acts like a trash can. So remember, exocytosis would be something that comes from the Golgi apparatus that needs to now leave in a vesicle and essentially needs to sort of pinch off to then release. This is oftentimes large waste or secretion and is released with vesicles. Okay, let's take some time and try to complete a question. Take a moment and read this, and then once completed, make sure you select your answer. All right, now that you're done reviewing the question, it says which one correctly describes facilitated diffusion. Did you select the correct one, which was answer choice C? If you did, good job. Let's review the other choices. It says it requires energy to form ATP. Well, ATP would be active transport. Remember, facilitated diffusion is a type of passive transport. Therefore, A would not be a great answer. B says it involves some sort of movement of water molecules. Remember, water molecules means we're talking about osmosis. And while that sort of falls under the category of passive diffusion and has some of the facilities similar to facilitated diffusion, remember when we're talking about facilitated diffusion, we're wanting to select the best choice answer. And so this would be a maybe for me until I read the further choices. Remember, for AP Biology, you're not expected to find all the answers that are correct, but which one would be best described. So this one would be a maybe before I look at answer choice C, which basically says it allows to move down their concentration gradient, which is what facilitated is, and then we're utilizing some sort of protein. So that's why C would be the best answer compared to B, because B would be best chosen for something like osmosis. Then, if you look at answer choice D, it involves engulfing large particles. Remember, that's describing engulfing into the cell, right? And when we're talking about into the cell, that means endocytosis with the process of engulfing large particles. Endocytosis is also a part of active transport instead of passive transport, so that's why it would be incorrect. So the best answer choice here would be answer choice C.
Appreciate you riding along for our cosmic journey. If today's mission brought you the knowledge that slaps, make sure to hit that like button to boost that signal. Subscribe if you're locked in for our next launch. And don't miss the link tree. It's got all the goods for AP Bio, PSAT Math, and SAT Math. Before I sign out, make sure you're all in one orbit and diving in. Killing out. Till next time.